Okay, so this is chapter 14, part 4. We're still talking about DNA structure and function. And in this part of the chapter, our focus is going to be on how DNA is replicated. And so we learned in the previous part of the chapter, part 3, that DNA is replicated prior to cell division and that it happens via the semi-conservative method. And so here we're going to talk more about specifics of how it's replicated. And so to start off, I'm going to tell you that when we talk about this, we're going to talk about how DNA replication occurs in prokaryotic organisms. So for example, like bacteria, like E. coli. And then after we're kind of done looking at this in prokaryotes, we will also talk about it in eukaryotes. Okay, and we'll talk about the many similarities that eukaryotes share with prokaryotes as far as this process occurs, and then the few differences. And so, why do we do this? Well, because this process was discovered in prokaryotes, and it's just easier to study this process in prokaryotic organisms. So, bacteria make great model systems because they have a bunch of features. So, number one, they only have one chromosome. So they have one circular piece of double-stranded DNA. Occasionally, they'll also have a few small circular pieces of DNA called plasmids. But again, the main chromosome of a prokaryote is going to be this one double-stranded DNA. And just so that you know, that double-stranded DNA molecule doesn't have proteins associated with it, so it doesn't have histones. So again, a little bit easier to study replication. What else? The genome, it's small. Because there's only this one chromosome, it has a small genome, and it's replicated very rapidly, very quickly, especially compared to eukaryotic organisms. It's also really easy to establish mutant organisms. So if we create a mutation that knocks out a protein, we can study how that affects replication, and therefore we can learn about the role of that protein in the process of replication. So easy, very easy to establish mutants. Replication also only starts at one region in prokaryotic organisms, and that's going to be called the origin of replication. In eukaryotes like us, we have many. Okay, and again, I'll talk more about eukaryotes after. So overall, when we go through this process, we're going to be mentioning a handful of enzymes involved in the process, a handful of proteins, which in this case are also enzymes, involved in this process. I want you to understand that there's a whole lot more to this process. There's more enzymes that I'm going to mention here. But again, we don't need to go that deep into it. So we're going to keep it, relatively simple. So we're going to cover the process, kind of an overview, and I'll introduce you to some of the enzymes involved here, but again, not all. Okay, so I just want you to realize that. Okay, so here's a list of some key proteins. This is only listing 3. There will be more that we'll add to this list, but we'll start off small. And so the first protein on our list here is called helicase. Okay, helicase. Notice the ending ASE. It's definitely an enzyme. Helicase is going to be responsible for binding to the origin of replication, which is where replication starts. And it's going to unwind the DNA. Okay, so it's going to take it out of that helical form. It's going to be nice and straight. And it's also going to unzip the DNA, which means it's going to cause the hydrogen bonds between the bases to break. And so it's going to expose single-stranded DNA. It's going to form what's called a replication bubble, which is composed of 2 replication forks, which I'm going to show you on the next slide. The second protein that's going to be important here, this is not an enzyme, it's just a protein. These are going to be called single-stranded binding proteins, also known as SSBs. And these are pretty important. They're going to bind to the single-stranded DNA, and they're going to prevent the DNA from re-annealing, which means sticking back together, right? We don't want the DNA to just, be opened and then boom, go right back together. We want it to stay separated so that it can be copied. Okay, and so the single-stranded binding proteins bind to the single-stranded DNA and prevent it from going back together. That's what re-annealing means. So we want to prevent those hydrogen bonds from reforming between those bases. And the third enzyme on here is one of my favorite enzymes. It's called topoisomerase. So again, a protein that also has enzymatic function. Its job is to prevent overwinding of the DNA double helix. And so, every semester I say this, and as the years pass, I've got more and more students who don't know what I'm talking about. So hopefully you get this. If you've ever been on a landline phone that has a cord to it, one of those spiral cords, I used to sit for hours as a kid. I'm holding that, and I would take the cord and I would, kind of straighten out that helical structure. And when I did that, down farther from where I was doing that motion, that straightening out, there would become these big tangled knots. Okay. The same thing could happen to your DNA, right? We're straightening out this short stretch, but down farther from that, it can end up getting super coiled. And we don't want that to happen. So this enzyme is going to prevent this overwinding, this super coiling of the DNA. And so if it starts to get some tension, some stress, then this topoisomerase will actually break the DNA, get the kinks out, and put it back together. So it prevents supercoiling and knotting of the DNA. It's a pretty cool protein. So let's take a look at these three proteins. Again, two are enzymes. So in blue here, I have my double strand of DNA, and right here it's open. Okay, and then I got more double-stranded DNA here. Okay, notice the 5 and the 3', right? Okay, that's going to become, that's important. DNA is going to be unwound in both directions, so this way, as well as in this direction, that way. Okay, here is the origin of replication. And again, in prokaryotes, there's just one origin of replication. Replication starts there, and then, it goes around the entirety of the molecule until it's finished. So the origin of replication is where this brown triangular enzyme originally bound to, and that enzyme unwound, and now it's unzipping the DNA. So it's heading into this region here, which is called the replication fork. And as it moves in, it unzips the bases. Here's a second helicase molecule. It's going into this replication fork. This whole opening is called the replication bubble, which is composed of two forks, one here and one here. So again, helicase is the brown triangle. Its job is to unwind and unzip. Now, when the DNA is single-stranded, like it is here and here, it could just go right back together. To prevent that are these little green proteins, which are called SSBs, or single-stranded binding proteins. Now, when the DNA is being unwound and unzipped, we could get supercoiling downstream. And so this purple enzyme here, topoisomerase, is going to prevent those supercoils from forming. Okay, so that's how DNA replication really gets started. Here is the next enzyme, which arguably is the most important enzyme of the process, is DNA polymerase. And there's actually, so in prokaryotes, there's 3 types of DNA polymerase. And we'll see later that in eukaryotes, there's a slew more. There's more than just 3. So DNA polymerase is also called DNA pol. So you'll hear me refer to that in short. There are 3 different types in prokaryotes. Okay. DNA polymerase 1 is going to be responsible for removing something called the RNA primer and replacing it with DNA. And we're going to get to that in just a second. DNA polymerase 2 is involved in DNA repair. Okay, and again, we'll get to that as well. But the third type of DNA polymerase is really the most important one right now. So DNA polymerase 3 is the main enzyme of replication. The job of DNA polymerase 3 is to read the single-stranded DNA template that was just unwound. And as it reads it, it's going to lay down and add complementary base pairs, complementary nucleotides. And it's going to add always in the 5' to 3' direction. And so in order for DNA polymerase 3 to work properly, there has to be a free 3' hydroxyl group. Okay, that's a problem. So if I go back a slide, helicase is unwinding and opening, and so we've got all this single-stranded DNA here and here that's ready to be copied, but DNA polymerase can't just come in and start copying. It can't work off of just a template. It needs the process to actually be started first, and then it can work because it needs a free 3' hydroxyl group in order to function properly. And for whatever reason, this is how it evolved. So there's another enzyme that's also really important, and that is called primase. And this is pretty strange. Primase is going to be an enzyme that's going to come in and start the process. And what it does is it lays down a short stretch of complementary bases, but it doesn't lay down DNA. It lays down RNA, which is very strange. You know, RNA is not in DNA, okay? It does this, again, to initiate the process. If there's a short stretch of RNA, then DNA polymerase can come in and it can work because now it has a free 3' end. So let me show you this. So now in orange, we have our DNA. So we've got one strand here running 5 to 3, and this strand here running 3 to 5. So our DNA is nice and anti-parallel. The bases would be here in the middle, just like a zipper. Topoisomerase is preventing overwinding, and it's going to relieve any tension, right, by breaking the DNA, untensing it, and putting it back together. Helicase is unwinding and unzipping. And here we've got our single-stranded binding protein, making sure that the two don't go back together. Okay, so now we've got primase. And so what primase is going to do, it's going to bind to the single strand of DNA, it's going to read the bases, and it's going to lay down a short stretch of what we call RNA primer. Okay, this RNA primer is going to be relatively short, only about 5 to 10 nucleotides in length. And it's laid down in the 5' to 3' direction. So right here where this 3' is, you should know that this is a sugar. This is the sugar phosphate backbone. This is the 3' carbon of that sugar is now free. And so if that is present, DNA polymerase can come in and start working. Okay? And so DNA polymerase comes in, DNA polymerase 3 is going to come in, and it's going to start right here at the edge of that primase, or sorry, the edge of the primer, and it's going to start to lay down complementary bases. So it's going to read this single strand of DNA, and it's going to lay down the appropriate bases. So again, A to T and C to G. And it can just kind of keep going, on this strand at least. It can keep going into the replication fork. But we're going to see on this strand there's a little bit more of a problem, because the DNA is going to be produced in the opposite direction, which is away from the replication fork. So we'll talk about that in just a second. Now, how about this RNA that's inside of our DNA? That can't be. So what's going to happen after the process starts, after DNA polymerase 3 gets going, what's going to happen is the RNA primer is actually going to be plucked right out. It's going to be removed, and DNA polymerase 1 will come in and it will fill in the gap. And so it's going to actually replace the RNA primer with DNA. Okay, here is another enzyme that's very important. It's called ligase. And ligase will come in and it will make sure that all of the backbone, the sugar phosphate backbone, is nice and sealed up. Okay, so it's going to, wherever the RNA came off, there's going to be some spaces left. And so ligase is going to come in and make sure everything is covalently bonded together in the backbone. Okay, so that's, again, how we get started. Okay, so now this is where it gets a little bit tricky. So the DNA is read by DNA polymerase 3 in the 3 to 5' direction and laid down in the 5 to 3' direction. So DNA polymerase 3 always needs a free 3' end. Okay, so I alluded to this problem a little bit just a second ago. Because the two strands of DNA, the template strands, are anti-parallel to each other, and because of the way that DNA polymerase 3 works, it can only work in one direction, 5 to 3', there ends up being one strand that's read and copied without a problem. It goes right into the replication fork. That's going to be copied continuously. But the other strand is going to be copied in a fashion which is called discontinuous replication. And so I want to go over both of those next. Okay, so this is the easy part, okay? the strand that gets copied continuously, no issues, is going to be called the leading strand. Okay, so let me show you in this picture. This is a different, the other replication fork, so a little bit different from the previous picture. So again, I've got in blue my original template DNA. I've got 2 strands of it, and I've got new DNA being laid down. Okay, and so again, DNA can only be laid down by DNA polymerase 3 in the 5' to 3' direction. So this top strand being made here, this little short stretch of red is the primer. Once that primer is laid down, DNA polymerase comes in, DNA polymerase 3, and just starts working away, right? Here's a free 3' hydroxyl. It binds there. It starts to add bases, and it just keeps going and keeps going and keeps going. Helicase will unwind this and unzip it, and this strand is just going to keep going till it gets to the end of the circle or the end of the linear chromosome, whatever we're looking at, okay? So that's called the leading strand. It can just keep going continuously. DNA polymerase 3 doesn't need to stop at all. It just goes and goes and goes. Okay, again, this primer here eventually does get removed. The little gap gets filled in by DNA polymerase 1, and ligase comes in and seals the backbone. We already know that. How about this other strand? And so let me just go to the next slide. The other strand is the one that gets copied discontinuously. Again, this is all because of DNA being anti-parallel, as well as DNA polymerase only being able to operate in the 5' to 3' direction. And so again, this is called discontinuous replication. It's not going to be continuous. It doesn't just keep flowing. It has to start, go, stop, start, go, stop. So there's all these little breaks. We call the strand that's being produced over here discontinuously, we call it the lagging strand because of all these starts and stops. Okay, and so what's going to happen is, where's my pointer? So the DNA was originally probably only unwound to about here, okay? Primase came in, it laid down a primer, which allowed DNA polymerase 3 to come in and start to lay down bases in this direction. And then our DNA got unwound some more, all the way to here. And so now this stretch of single-stranded DNA, the template, needs to be copied. This is going in this direction. So what has to happen is another primer has to be laid down. DNA polymerase has to come back in and fill this in. And then this will be unwound more. And guess what? Another primer has to be laid down. DNA polymerase 3 can then operate, right, and fill it in some more. And so we have this discontinuous, this start and stop. And then what ends up happening is when we take the primer out, we end up with a whole bunch of gaps. Okay, and those gaps, they do get filled in by DNA polymerase 1. Ligase comes in and seals the backbone. But all of these little short stretches of DNA, they get a special name. The name is after it was actually a husband and wife team of scientists that discovered these. They're called Okazaki fragments. And so Okazaki fragments are only produced on the lagging strand, not on the leading strand. Okay, so let's show you this in total. Okay, so if this is my replication bubble, right here, this whole opening, okay, the origin of replication is right here, and replication is occurring in this direction into this replication fork. It's also occurring in this direction into this replication fork. This is called bidirectional replication. Okay, it really speeds up the process, right? So if I have a circular piece of DNA and I start at the top, by the time I get to the bottom, I'm done. Okay, same thing on our chromosomes. You have a linear chromosome, you have an origin of replication. There's actually many, but let's say it's in the middle, and you just go towards the ends and you're done. It's not starting at one spot and going all the way through for a linear chromosome. Okay, so take a look at this replication bubble. Notice to the left, or sorry, let's do to the right. To the right of the origin of replication, it's the same thing that we just looked at. So this is a single replication fork right here. Here's our leading continuous strand, and these are our lagging strands over here where we have our Okazaki fragments. But if we go to the left of the origin of replication into this replication bubble, it's actually the opposite. The bottom strand now is the leading, and the top strand is the lagging. Okay, so it's not like one strand is, one and one is the other. It all depends on where the replication fork and which direction replication is occurring in. Okay, so again, it's really important to remember that DNA polymerase III can only lay down bases in the 5' to 3' direction. Okay, and so that's why we end up with a leading and a lagging strand. Okay, so on the next slide, this last one is just a nice summary of replication. Okay, so again, this is the list that you should kind of go through once you feel comfortable. It has all the major players on it, all of the enzymes. So we've got helicase, it unwinds and unzips at that origin. Replication forks are produced. They're going to be extended bidirectionally. Single-stranded binding proteins come in and prevent the DNA from sticking back together or re-annealing. Topoisomerase makes sure that there is no supercoiling or tension on the DNA. Primase lays down a short stretch of RNA primers to start the process. DNA polymerase III actually puts together the DNA bases to form the new DNA strands. Elongation happens on both the leading and the lagging strand, but remember that the lagging strand has Okazaki fragments. The RNA primers are taken out. The gaps are filled in by DNA polymerase I, and the gaps in between fragments are sealed by DNA ligase. Okay, so that is the process of DNA replication.